Welcome. Today we are going to learn about the alpha complementation and the blue-white selection. Going towards first the alpha complementation. Alpha complementation, it has been first observed into the E.coli M15 mutant. It was a mutant for the laxate gene which was unable to code the 11 to 42 amino acid residues. Okay, but when in vitro the cyanogen bromide it has been cyanogen bromide peptide when it has been added. This cyanogen bromide which is derived from the 3 to 92 amino acid residues of the beta galactosidase, then it has been observed that the activity of the beta galactosidase it has been restored. Means what? When in trans there is the providing of the missing part of the beta galactosidase, then there is a restoration of the beta galactosidase activity. And here that missing part, it is the alpha peptide. That is the N terminal of the laxate gene. When the N terminal of this laxate gene, it has been put on the plasmid and allow the plasmid to transform the M15 mutant, E. coli M15 mutant. At that time, it has been observed that in vivo also, there is a restoration of the beta galactosidase activity. Okay, means what? The beta galactosidase, it has the two separable domains. Okay, and here it is little bit about the beta galactosidase. Beta galactosidase, it is a tetramer of the 4 identical monomers, and each monomer it is of the 1023 amino acids. Okay, so just now what we have seen that the laxate, it is a laxate mutant. Okay, M15 laxate mutant, it is a mutant at a N terminal. That N terminal, it is called as the alpha peptide, and the remaining part, okay, remaining larger part, it is called as the omega peptide. Okay, so here when there has to be the restoration of the beta galactosidase activity, these all 4 tetramers, okay, all 4 monomers, it has to be come together and it should form a proper functional unit, that is a proper tetrameric form which is work as a beta galactosidase. And to have that, okay, to have that functional beta galactosidase, what is required? This alpha peptide it is required. Okay, so here, okay, the smaller part at N terminal, it is a alpha peptide, and the larger part at a C terminal, it is a omega peptide. Now here the omega peptide plus the alpha peptide, it is going to give you the functional beta galactosidase. Omega peptide, it is also called as the alpha fragment acceptor, and alpha peptide, it is called as the alpha fragment donor. Alpha, it is also called as the N terminal fragment. Omega, it is also called as the C terminal fragment. Okay, so here when a bacterial cell, that is the E. coli cell, which is defective into the laxate at the N terminal, that is which is unable to synthesize the alpha peptide, when it has been provided in trans with the alpha peptide, and it is allowing the restoration of the activity of this beta galactosidase, then it is called as the alpha complementation. Okay, that is the alpha peptide, it has been provided to complete the structure of this beta galactosidase. So the beta galactosidase, it is going to be a functional beta galactosidase. And as this has been discovered that there will be the 2 fragments, they are going to be there, and 2 fragments, it can be located on the 2 different places, and in trans, when they are present, they are going to come together and it is going to form a functional beta galactosidase. So this, it has been utilized like a marker. Okay, this it is going to be utilized as a marker, and here the vector, here the example of a vector, it is a pUC18. And in this, okay, in this pUC18, what you can see that there is the laxate gene. Okay, this laxate. And this laxate gene, it is encoding only the alpha peptide. Okay, what it is encoding? It is encoding the alpha peptide, that is the N-terminal portion of the beta-galactosidase. Only it has been encoded by the laxate gene. That is, in this laxate gene, there are only about the 146 codons. Okay, and here there is a ampicillin resistant gene. There is the origin of replication. Here you can see there is a MCS. MCS, it is a multiple cloning site. Okay, and here these are the restriction sites which are present into this MCS multiple cloning site. And this laxate, it is under the control of the LAC promoter, and it has the binding site for the LAC repressor. Okay, there is a LAC-R binding site, LAC repressor. So the LAC repressor, it can bind here, and it is going to inhibit the expression of this laxate gene. Means what? This laxate gene, this alpha peptide, it is inducible one. Okay, the expression system which is maintained here, it is a inducible one. Okay, induction, it is present here, and that induction, it is done with the help of the IPTG. Okay, so here it is the example. There are number of examples which are utilizing this alpha peptide or this defective laxate gene on a vector, and that examples, it is the pUC18, lambda zap, then there is a pBlue script, then there is a pMBL18, pMBL19, and in all that there is what? The laxate, it is with whom? It is only encoding the 146 codons. Okay, now this is regarding the vector. Okay, but a vector, it is a defective. Okay, it cannot synthesize the omega, but what it can synthesize? It can synthesize the alpha. So how should be a host then? Okay, how should be a host? The host here, it has the mutation. Okay, it has a deletion mutation at the N-terminal, and that deletion mutation, it is called as the laxate delta M15 deletion mutation. The deletion mutation which is present into the host, it is called as what? It is called as the laxate delta M15 deletion mutation. Okay, which cannot synthesize the N-terminal amino acids about the 11 to 41 residues of the beta-galactosidase. Okay, from the N-terminal, it cannot encode the 11 to 41 residues of the amino acids. Okay, and in this way, there is a host, and the examples of the host here, it is the E.coli MG1655, E.coli DH5 alpha. And here in the E.coli DH5 alpha, this laxate gene, it is constitutively expressed. And this, okay, the laxate gene, okay, this laxate gene which is present in the host, it is present over the genome. Where it is present? It is present over the genome. Defective laxate in the host, it is present over the genome. Okay, now going towards the alpha complementation. Okay, going towards the alpha complementation. Or you may call it as an blue-white selection. Alpha complementation used for the selection of the recombinant and the non-recombinant, that it is a blue-white selection. Now here the cloning the insert. Okay, for the cloning of the insert, you should have a vector. Okay, just now we have seen the example of the vector. It is the pUC18. Suppose this plasmid, it is a pUC18 plasmid, and this one, okay, this one, it is the laxate. Okay, laxate which is encoding what? Which is encoding the which part of the beta-galactosidase? Yes, it is the alpha peptide, and here it is the ampicillin resistant gene. Okay, next here we have to, okay, when you have to add an insert, you have to go for the restriction digestion, and for that purpose you have to use the restriction enzyme. From the MCS, you can select any one of the restriction site, and accordingly that you can select the restriction enzyme, and you can open up the vector. Okay, restriction enzymes, which are also called as the molecular scissor, it is going to cut the vector. Okay, and in this way there is a opening of the vector. Next, there will be the, okay, then you are going to add a insert, and after addition of the insert, okay, you have to go for the ligation with the help of the enzyme ligase. So when you are going for the ligation, you are going to observe that the insert, it is going to get ligated with the vector. Okay, in this way. Okay, this it is a vector, and this blue color, it is a insert. Okay, the insert, it has been ligated with whom? It has been ligated with the vector. That is, it has ligated in the pUC18 vector, and this, it is called as a recombinant vector. Okay, it is a recombinant DNA molecule. It is a recombinant vector because it is consisting of the insert. Or the second possibility, it is what? Okay, second possibility, it is of the self-ligation. That is, this vector, it can ligate to itself. Okay, the possibility, it is what? It can ligate to itself. So there are the two products it is going to be there. First one, it is a recombinant vector. Second one, it is the self-ligated vector. Okay, next, there will be the transformation of the E. coli cells. Now here it is a ligation mixture. In the ligation mixture, it is what? It is the recombinant. Okay, this one, it is a recombinant. It is a self-ligated. It is not ligated vector, and this, it is an insert molecule, and it is the it is a vector with a fragment. Okay, now that we don't want to get inserted into a vector, but this possibility, it is very lower one. Okay, so here these are the cells, these are the E. coli cells, okay, which are the competent cells. For the transformation, we need the E. coli cells, which are the competent cells, and these cells, okay, these cells, it may be it is consisting of, okay, it is consisting of the laxate, which is encoding which part of the beta galactosidase? Yes, it is a omega peptide. It is going to encode for the omega peptide. Okay. Now here, after transformation, what you observe, that you got the plasmids. Oh, sorry, you got the bacterial cells with no plasmid. Okay. There is no transformation in some of the bacterial cells. Some of the bacterial cells will get the unligated vector. Some, it will have the unligated insert molecule. It has been transformed into the E. coli cells. Okay, and in some, and there is a self-ligated, okay, self-ligated vector, okay, vector with no insert. In some, undesired insert with a vector, and here there will be the vector with a desired insert. Okay, so these many possibilities will be there after transformation. These many types of the cells you are getting. And now from all these 6 cells, okay, from all these 6 possibilities, what you have to choose or you have to select? You have to select for the vector with a desired insert. Okay, what you have to select? You have to select for this. Okay, you have to select for this. Okay, vector with whom? Vector with a desired insert you have to select. For that purpose, you have to go for the plating. Here, there is a growth medium. This, it is a first plate in which there is a growth medium, that it is a control one. Okay, and the second plate, it will have the growth medium plus ampicillin plus X-gal plus IPTG. Now, as we have taken the example of the pUC18, on the pUC18, there is an ampicillin resistant gene. So here we have to add the ampicillin into the medium. X-gal, it is a chromogenic substrate. IPTG, it is an artificial inducer. Okay. So, so here, okay. So here you can see here that okay, these types of the cells we have to eliminate. Okay, the no plasmid, unligated insert, okay, and unligated vector, okay, these we have to remove, and that it is going to be removed. Okay, that cells, they are going to be that cells, they are not allowed to grow when you are going to plate over this medium, because these cells, as there is a no plasmid, as there is a no plasmid, okay, as there is no plasmid in where in the first one and in this third one, no plasmid. So what will happen? It don't have the any ampicillin resistant gene. As there is a no ampicillin resistant gene, that says they are unable to grow on the growth medium consisting of the ampicillin. Okay, now here, okay, now here what we have to select for now, we have to select for this desired insert. Now just now what we have seen, okay, just now what we have seen, that the plasmid, it is going to provide the alpha peptide, and as the plasmid, it is going to provide the alpha peptide for the host. The alpha, it is going to combine with whom? It is going to combine with the omega, and it is going to form the functional beta-galactosidase. This it is going to be happen, okay, this it is going to be happen with whom? With the self-ligated, with the self-ligated, because when we are cloning, okay, when we are cloning into a vector, okay, we are actually going to disturb the lag Z gene. We are going to cut, okay, we are going to cut the vector, okay. This it is your vector, and by the scissor restriction enzyme we are going to open up. Means what you are doing, you are disturbing the gene, you are disturbing the lag Z gene, and in that when you are going to add your insert, at that time that lag Z, it is becoming a inactive one, that is, it is not going to synthesize the functional alpha peptide. Okay, so when there is a no functional alpha peptide, there will be no alpha complementation, and when there is a functional alpha peptide, at that time there will be the alpha complementation. So what do you think, that when you are going to see the alpha complementation in a self-ligated vector or in the vector which is a recombinant? Yes, yes, you are going to see the alpha complementation into the self-ligated. You are not going to observe it into the recombinant vector, because in the recombinant vector, okay, in the recombinant DNA molecule, there is a disruption of the lag Z gene, the alpha peptide, it is an inactive one. Okay, so on this, okay, on this growth medium, we have to select for the recombinant one, and for that purpose, there is a use of whom? There is a use of this X-gal, which is called as the chromogenic substrate. And this, it is a substrate, okay, this, it is a substrate for the enzyme, and that enzyme, it is the beta-galactosidase enzyme. Beta-galactosidase, it is going to act on the X-gal, and it is going to give the blue colored complex. Okay, so here when there is, okay, whenever there is a alpha complementation, okay, when there is the alpha complementation, at that time there will be the functional beta-galactosidase. Okay, then there will be the functional beta-galactosidase. Means when there is a self-ligated, okay, when there is a self-ligated vector, at that time it is going to synthesize the functional alpha peptide, and that alpha peptide, it can complement with the omega peptide, which has been encoded by whom? Which has been encoded by the host, and then it is going to combine, it is going to form the functional beta-galactosidase, and that beta-galactosidase can act on the X-gal, and as it is going to act on the X-gal, it is going to give you the blue colored complex, and as there is a blue colored complex, the colonies that you are going to observe on the plate, on the growth medium, that colonies, they will be the blue colored. Okay, this one, okay, this one, these all the blue colored colonies. Okay, the blue colored colonies that you are observing, all they are the non-recombinant. Okay, all that they are the non-recombinant. And what you can say about the white colonies now? You have cloned the gene. Okay, you have cloned the gene into the pUC18 in the lag Z gene. Okay, so as you have cloned into the lag Z gene, and it has been disturbed, do you think that the alpha peptide, it is going to be a functional one? No, it is not functional. So if it is going to form the functional beta-galactosidase, Yes, no, it is not going to form the functional beta galactosidase as the alpha peptide. It is a non-functional one. There is no presence of this alpha peptide. It cannot complement with the omega peptide as there is a no alpha. How it can complement with the omega? Okay, so there is a no. Okay, there is a no functional beta galactosidase in the host cells in which there is the presence of the recombinant vector. That is a vector with a insert. Okay, so here you are going to observe that the colonies, they are of the white colored. Okay, as there is a no beta galactosidase, okay, no functional beta galactosidase, it cannot act on the X-gal. As it is not going to act on the X-gal, there is a no blue colored complex. No blue colored complex means what? The colonies, they are white in color. So in this way, okay, in this way, on the single plate, okay, on the single plate, you can select for the recombinant and the non-recombinant. You can select the recombinant colonies. Okay, white color colonies, it will be a recombinant, and blue colored colonies, it will be the non-recombinant. In this way, by the use or by the application of the alpha complementation, you can select for the recombinant and the non-recombinant, that it is called as the blue-white selection. Okay, I think you may have cleared the idea. Okay, if you like my video, please subscribe my channel and also share my videos with your friends so the knowledge it is going to spread. Thank you.